All right. Seems good.
Where we were both studying mechanisms of metabolic dormancy, I was working on this fish that I'm going to talk with you about today, and Frank was working on brine shrimp at the time. So he went definitely to more complicated organism later on. So since this is the first talk of the workshop, I wanted to give you just a brief overview of epigenetics and some of the mechanisms that are used by organisms to convey epigenetic information. Because it'll add context for the different ways that I'm going to show you epigenetic regulation works in this particular model. So what is epigenetics? It sounds like an easy question, but the definition of epigenetics has changed drastically over the last 50 years. And in fact, so much so that they had a meeting in 2008 at Cold Spring Harbor, I think it was, or Mount Desert, I can't remember, one of those two. Where all of the folks at the time who were working on it came together and came up with a consensus definition, which is an epigenetic trait has been defined as a stably heritable phenotype resulting from changes in a chromosome without alterations in DNA sequence. So it's kind of a crappy definition, as all definitions that come from consensus are, but because it's just way too broad to be useful. Sort of a useful working definition is a heritable change in gene expression within a cell or a whole organism without an underlying change in DNA sequence. So this allows us to look at changes in cell lineages during the lifetime of the organism and all of the daughter cells or those cells within a body, but also the potential for transgenerational communication of those changes. Okay. And just the concept of epigenetics is that all of these things that we encounter in our environment, so heat stress, hormones, pathogens, our microbiome, toxicants, they all get incorporated into changes in the genome that are not sequence-based, but some other way they're coded in the cells. And the true expression of the genome is an interaction between the genome and these epigenetic changes that we're going to talk about today, which leads to changes in gene regulation and then therefore phenotypic changes. Okay. So I study fish, so I had to find a fish-specific figure here. But the one way to think about this is that the genome's there, and then there are all these environmental stimuli that you have to deal with on a daily basis as a typical organism. But there's also a bunch of endogenous things that might change. Hormonal levels might change, or you might have an altered metabolism for some reason, either through a mutation that's completely unrelated with the epigenetic change, or through changes in the environment that work through your endocrine system, for instance. And that causes changes in these modifications to basically chromatin structure for the most part, but also in RNAs and other things. And that leads to these similar changes in gene expression that change your function or your phenotype. And so it's basically a way, I like this because it's basically a way to make your genome context dependent, right? There's no way that a genome is going to be perfect for every situation. And so you need some plasticity or flexibility in that genome. We know now of many of the mechanisms that are used to transmit epigenetic regulation. And if we have known all of this when Waddington was started, he wouldn't have been ignored and sometimes even ridiculed for his ideas. So the first sort of mechanism that most people think about when you think about epigenetics is DNA modifications and DNA methylation. Okay, so we can actually methylate the cytosines in CG or CPG pairs within the genome, and we can use that to change the activity of a stretch of chromatin. And it sounds simple. It used to be when I first learned about this, it was simple. If you methylate DNA, you silence DNA, and now we know that that's not true. Methylating DNA has all sorts of different effects. Sometimes it alters splicing, sometimes it alters it turns the gene off, like if it's in the promoter, but if it's in the gene body, it can actually alter splicing, or it can actually increase gene expression. So you have to know a lot about where those methylation changes are happening in order to make sense of it. There's also what's come to be known as the histone code. So the histones that wrap up and package the DNA have these N-terminal tails that stick out. Those can be modified post-translationally by, I think that the last review paper I saw, there was over 500 different modifications that could happen to the histones. And by changing those histones, or there's also histone variants in some species, you can actually change the availability of the DNA to interact with transcription factors and to be expressed. So we've got DNA changes, we've got changes in the histones. We've also got non-coding RNAs, which is an area that's exploded in the last like 20 years. We've got long non-coding RNAs that can bind to proteins, alter expression. We've got small non-coding RNAs like the microRNAs or pRNAs that actually either end up signaling for the degradation of an mRNA or blocking its translation is the most likely route for that. And then we also have RNA modifications. So for instance, your tRNAs are highly modified, and many of your small RNAs are highly modified as well. So those are the basic mechanisms that we know that this epigenetic regulation is mediated through. But I also just like to put this little figure up because it gets very complicated very fast. There are very direct routes to altering this epigenetic landscape that a lot of people end up talking about. But there's also indirect routes, right? So you can have a route that directly changes. You express a protein or a histone deacetylase, for instance, that will change the availability of some stretch of DNA to transcription factors. You can also have that same thing happen indirectly through dysregulation. So there's a huge body of work out there on cancer. And in cancer, you sort of get an indirect dysregulation of your epigenetic landscape, which causes a lot of problems. This is also sort of a route for environmental exposures to cause changes in the epigenetic landscape as well. All right, so back to Waddington. So Waddington was a super smart guy, and he had come up with this concept, and I'm putting this in context of development now. So think about all this epigenetic information that we can encode in our cells, and then think about how, if you change the epigenetic landscape during development, it can have lifelong effects, basically. And so Waddington had come up with this concept of an epigenetic landscape, which you see here, where you've got valleys and ridges. And he sort of thought about development as navigating sort of the path of least resistance. And in an ideal world, like let's say you're in constant conditions, everything's great, you've got everything you need, you always take this route and you end up with what we call a wild type organism. If you're perturbed or stressed during development, which every organism is basically at some point in time during their development probably, then you sort of get pushed away from this ideal route. But if the buffering mechanisms that you have are strong enough, you get forced back into the normal route and you end up still at wild type. If these perturbations are too much, basically, you get pushed out of the normal valley or trough of wild type into a new one, and you actually can have an altered phenotype. So this was what Waddington was talking with people about, and they're like, oh, this is Lamarckian, you can't inherit this stuff, and the truth of the matter is you honestly can't. And the hard part for people to swallow was this idea of assimilation, that they could swallow that you might change a phenotype because you can use a mutagen or something to cause abnormal development. But the fact that that abnormal development could then be transmissible and heritable, people just didn't buy. And what we found out since then is that the very mechanisms that sort of buffer the developmental program, like heat shock proteins like HSP90, if they're swamped, then there's all this cryptic variation that can be expressed and it can be fixed, because only those organisms that have that cryptic variation survive, the rest of them don't. So, and that was sort of when that started happening, that was work that was done in the 90s by Susan Lindquist and her group. So now the way that we look at these landscapes is that most organisms develop along this sort of normal route. And then if you stress them enough, you can push them into these other non-standard routes, but they can become fixed if for instance, the environment stays the same, okay? So We still don't really understand a lot of the mechanisms that allow these to become heritable, but for instance, in zebrafish, changes in methylation of the genome can last, I think, I've seen at least 10 generations now for some studies. So we do know that many species keep their methylation patterns for long periods of time. And actually, the mammals are really weird in terms of their methylation because we erase our methylation patterns during development and then regenerate them. But most organisms don't do that. Fish don't do that, for instance. Okay, so with that backdrop of epigenetic landscape, I'll introduce you to my study system. So I work on annual killifish, which live in ephemeral ponds in tropical environments that are highly seasonal. Right? So these environments are typically wet, have a wet season that might last 2 to 3 months, and then they're dry the rest of the year. And this is just a temperature, graph of temperature in one of the ponds that my fish live in. And you can see that every day, each one of these is a daily cycle. The fish are going to go through a 15 to 20 degree temperature change every day. So just their normal life. For us, a 4 or 5 degree fever makes us feel really awful, and much more than that, and you're dead, right? For these guys, 4 to 5 degrees, they're just waking up in the morning. They also, along with that change in temperature, drives all sorts of other changes. pH, oxygen concentration goes from hypoxic in the morning to hyperoxic, almost 300% saturated in the afternoon, because you got all these algae growing in the tropical sun. And the embryos, which are put into the substrate, are almost immediately anoxic. This sediment PE indicates that the redox potential is way towards no oxygen or reduced and not oxidized. So I became super interested in trying to figure out how do you go from a single cell egg to this multicellular organism that can survive in an environment that's this variable on a daily basis, and then put on top of that seasonal patterns and all sorts of other things. And I came from sort of a biochemical physiology adaptation background, so I kind of view this as these organisms are sort of on the knife edge between having good enough physiological buffering systems to allow development to occur, but also having enough mechanisms where they can use physiological induction, epigenetics, phenotypic plasticity, to adapt to the environment that they're in. Okay, so but you can't go too far in either direction, or it's the end of the line for you. All right, so annual killifish, I call them vertebrate extremophiles because they can survive things that few other vertebrates can. They can survive 150 days without oxygen. They have a brain and a heart. The heart stops, their brain goes dormant, and they can sit there. Take them out 150 days later, put them in oxygen, the heart starts up over the next 12 to 14 hours, and they just continue development. They can survive for, I think the record is 600 and some odd days at 75% relative humidity, which is, so that's almost 2 years without access to water, liquid water, and in dehydrated conditions, right? 75% relative humidity is the equivalent of being in about 2x sea salt, okay? They're super pretty fish. There are people all around the world, there's killifish associations in almost every country. And because the embryos can survive in dry substrate, they just mail them to each other all over the world. It's pretty cool. But you don't see them in aquarium stores because they only live for about 2 months to a year. And it's hard to explain to your kid why their fish died, right? Nobody wants to do that. I did this for my son. I brought a killifish home, but I would just, every time I thought he was going to die, I would take back and bring a new one in. So we have the longest lived killifish in the world, Gil, who lived for like 6 years. So. All right, so here's a typical pond. This is actually in southern Mozambique in Africa. The species that live in here are Nothobrachus. Here's a pond from northern Venezuela in the Maracaibo Basin. This is the pond right here. And the species I study, Ostraconeus linnaeus, lives in these ponds. So we're going to go and look at right here at the top of South America. Interestingly, if you're an evolutionary biologist, the group of annual killifish that are up here and down here in South America theoretically evolved this life cycle I'm going to show you independently. And these species in South America theoretically evolved them independently of the ones in Africa. Yet their life history is exactly the same, almost undistinguishable. So there's Ostraconeus linnaeus, there's the male, that's Gil, and there's a female here. And this is a typical rainy season habitat. Looks like a nice place to live, doesn't it? The water is hot and nasty and gross, and probably in this little pond that's not much bigger than a bathtub, there were probably 500 or 600 fish in there. And there's a dry season habitat. So obviously most people don't go to a pond like that and look for fish. So how do they do it? Well, they've evolved this life history that includes 3 different stages of diapause. And this is pretty unique. Most organisms that have dormancy stages typically invest in one stage. There's one spot, sometimes 2, but these fish have 3. So if we start over here with their life cycle, when there's a pond and the fish, they spawn daily. So they'll spawn their eggs into this really thick clay mud that's at the bottom of these ponds. And we know now that during the rainy season, all the embryos stop at diapause 1. Diapause 1 is before there's even an embryonic axis formed. It's just a whole bunch of embryonic stem cells in there, basically. Once the ponds dry up, they quickly develop into diapause 2. This is the most stress tolerant of the diapause stages. It's got I don't know if you can see the embryo here, but it's got all the hallmarks of a vertebrate embryo right here. It's got a brain, so there's actually a forebrain, midbrain, and hindbrain. It's got eyes, there's a heart up here, and then there's all these balls of somites on the side that are going to make up the body of the fish. These guys can survive, like I said, for hundreds of days or years without oxygen or water. Once the ponds start to fill up again, these guys start to develop, and they go to diapause 3, which is essentially a fully formed fish larvae. When these things hatch, they eat immediately. So they're very precocious hatchers. And they will eat each other and anything that you put in there with them. So 3 different stages of diapause, and that helps them to extend their developmental period for the 6 to 9 months that there's no water on, basically. All right. So what do we know about the control of this? So a lot of the control of this life cycle is epigenetic, what we're finding now. So we know for one thing that if you're a young female, you're going to produce, and this is kind of hard to see with this, whatever that thing is there, you're going to produce more embryos that don't go into that diapause 2. Oh, I forgot to say that. Let's go to this. Females can produce embryos that take this trajectory, or they can produce embryos that don't go into any of these diapauses and just directly develop, okay? So that's what we see here. Those embryos that directly develop are called escape embryos because they escape diapause. I hate the name, but John Worms in 1972 called them that, and so to try to be consistent with the literature, we always call them that. So you can either see, sometimes I'll call it bypassing diapause 2 or escape embryos. Anyway, early in a female's life, she produces a lot of escape embryos, and then the older she gets, the more and more diapause embryos that she'll produce. However, there isn't a single female that I've ever monitored that produces 100% one phenotype or the other. There's always some mixture in there. So, and we also know though, that if we manipulate the embryonic incubation temperature, we can override whatever the female has programmed in. So if we take any embryo and put it at 20 degrees Celsius, 100% of them will go into diapause 2. If we take any embryo and put it at 30 degrees Celsius, just 10 degrees more, 100% of them will not go into diapause 2. And you can titrate any percentage you want, basically, in between, by about 22 to 28 or whatever is the span where you might see sort of a constant increase in escape embryos across that. So we use this in the lab a lot to control the developmental trajectory of the embryos so that we can look at their physiology and their pathology. We also know, it's hard to see up here, that the bars that are this orange and brown colors, this is the wonder of graduate students. I don't know why she chose this color. That was awful. Anyway, if you just give them 2 hours of intense light and heat, that will do the same thing as putting them at 30 degrees for their whole developmental period. And it has to be during this window of about 10 to 15 days post-fertilization. There's a window where if you just give them a nice bolus of hot temperature or light, and light alone is enough to do it if it's bright enough, then they'll also escape diapause. So we sort of have some tools to manipulate the system, and we have ways that the environment can be integrated into the developmental program. And we've tried to exploit that in my lab here for the last little bit. So let's start by looking at what we know about the effective temperature on the developmental program. So what we did is we took embryos and we incubated them at 20 degrees Celsius and at 30 degrees Celsius. And we extracted total RNA from each of these embryonic stages that look morphologically exactly the same. You can't tell them apart. But we know that they're on different trajectories because if we leave them in there, they're going to eventually end up in either diapause or an escape embryo. And then we used RNA-seq techniques to compare the transcriptomes in each of these stages. Okay, and I've left off this work was done by Amy Romney when she was a PhD student in, I think we published this in 2018. So what we did was we extracted that RNA. I left off a whole bunch of slides to just shorten the talk, but we used RNA-seq, and then we used a bioinformatics pipeline called Weighted Co-Expression Gene Network Analysis, WGCNA. And all WGCNA allows us to do is group genes based on similar expression patterns, right? It's like doing a giant correlation matrix of every gene in the genome. It's not the best method in the world, but it does have some advantages, and we were able to exploit it as a tool, I think, pretty effectively. So what we did was we took the diapause embryos and we created this gene network from them. And from that, the program basically makes these modules, which it names different colors. I don't know why they used colors, but so each of these colors is a different module. So that means that's a group of genes that are all expressed in a similar manner. And you can see that over here. So this light yellow module is this one. And you can see that these are all on. Yellow is highly expressed, blue is lowly expressed, black is equally expressed. So that's all on in our first developmental stage, that dispersed cell phase right here. And then it's essentially off or low for the most part in the rest of the developmental program. So what we did is we took these modules, and then with this technique, you can ask the question, is this gene expression module conserved in two different conditions? And that's what we did. So we developed these modules for diapause, and then we asked, so this is all 20-degree data, and we said, is that module conserved at 30 degrees when we look at the 30-degree data? And people use this at first to compare developmental programs across different species like mice or rats or humans and mice to look for conserved developmental programs, which is super useful and super easy. We're just the opposite. We're like, I want to take all the normal developmental stuff and subtract it out because we know that that's not changing the developmental trajectory. We want to know the stuff that isn't conserved in these two areas. So that's what we did. So all of these modules that have little asterisks are modules that are different, not conserved, between the diapause 20-degree treatment and the escape 30-degree treatment. And as an example, and actually the one that got me the most excited, I just put this little yellow one up here. So there were only about 40 genes in this particular module. And one of them that came up as very important for constructing the network in the diapause was this DAF36 light, which is very central to the network here. But at 30 degrees, it's actually more peripheral, and there are other genes that seem to be important in the center of the network. But also important, if you look here, this DAF36 is one of the genes up here that's off or very, very lowly expressed in diapause and highly expressed in the escape embryos. So I got excited about this because DAF is actually a gene in nematodes, C. elegans, that controls dower formation, which is another form of dormancy. So I thought, wow, that's cool. Maybe we should explore this a little bit. So we looked at dower formation in C. elegans, and so just a quick overview of that. C. elegans is a nematode worm that it's a major model and everything, so we know a lot about it. We know in the lab if they're crowded or if they don't have enough food or if you heat stress them, that they can take this alternate developmental trajectory called dower. And they'll sit there, so they go dormant and they'll sit there for a couple of months. And then once conditions are conducive, they will enter into the developmental program again and then go through reproduction. So we know a lot about the molecular mechanisms that control this in C. elegans, and that DAF36 is the first enzyme in the biosynthetic pathway that creates these acids called daphrochronic acids, which are actually derivatives of 7-dehydrocholesterol. So they're a cholesterol backbone. They're not that different really from steroid hormones or other things. And it turns out that in worms, if you make a bunch of daphrochronic acids, they bind to DAF12, which is a nuclear receptor. So nuclear receptors are these ligand activated transcription factors, basically. So the estrogen receptor, the androgen receptor, corticosterone receptor, all of these things that we think of as being very powerful at mediating changes in phenotype, especially if you think about the sex hormones, estrogen and testosterone. So it's similar to that, but its ligand is this daphrochronic acid. So when daphrochronic acid is around, it binds to DAF12. That signals to the worm that things are good, life is good, let's reproduce. And so they go through direct development and reproduce. In the absence of daphrochronic acids, this DAF12 nuclear receptor then can interact with co-repressors, and it goes around and activates a whole different gene expression program that causes the worms to go into tower. Okay. So we have a hormonally controlled system in C. elegans that is dependent on the production of these daphrochronic acids through and mediated through DAF12, the nuclear receptor. Okay, so I got super excited about this because I'm like, okay, so we found this biosynthetic gene that is homologous to a gene in this pathway. Could it be that it's actually a similar mechanism in killifish and in nematode worms for dormancy, which is kind of wild because they are really, really different. And I don't even know what the divergence is between a nematode and a fish, but it's a long ways. Okay, so we decided, let's just look for all the homologs of these DAF genes in killifish and see what's going on. And so there's a couple of different pathways that can induce dower in C. elegans, but the one that's important here is the hormone one up here. And what we can see is that there are certain genes that are super important in inducing dower, and they're highly expressed in an interesting way in these killifish. And what was most interesting is that DAF12, its homolog in vertebrates, is the vitamin D receptor. Okay, and so we got super excited about that because now we've got the vitamin D receptor highly differentially expressed in escape and diapause in a way that's very similar to DAF12 in C. elegans. So we went headlong into looking at vitamin D and its role in regulating dormancy in these kind of killifish. So just a little background on vitamin D. I'm sure everybody knows all this, but just for the sake of clarity, vitamin D is made from cholesterol backbone, and the first step is to make 7-dehydrocholesterol, okay, and that's called provitamin D3. For humans, for most mammals, and for many vertebrates, that 7-dehydrocholesterol, when it's in the skin, gets converted to vitamin D3, cholecalciferol. That's the stuff that you take if you take a vitamin D supplement, through the action of either UV light or heat, and usually both. Usually you need both, right? So that's why, if you think back to when I told you earlier, like short bouts of intense light or heat are enough to drive them towards diapause, or sorry, towards escape. That could be because it's actually forcing the production of vitamin D in the lab. So then cholecalciferol, the stuff that you take in humans, it goes to the liver then through your blood, and it's hydroxylated at the 25 position by the 25 hydroxylase. And then that flows back into the blood to your kidneys, where you put 1-alpha hydroxylase on there. And that's the active form of vitamin D. So if a cell's going to respond to vitamin D, it's responding to 1-alpha dihydroxy vitamin D. Okay. And for the longest time, we've associated vitamin D with calcium homeostasis, right? Everybody takes it to battle osteoporosis and things like that. That's actually a minor part of what vitamin D does. It regulates so many things, and we're finding this like every day. Somebody's finding some new role for the vitamin D receptor in regulating different parts of cellular physiology. It's especially important in the immune system, but it's important in heart health and development and brain health and development for sure. Okay, so with this in mind, we did some experiments, and I apologize, this is a super busy slide, but the most important thing is to tell the difference between blue and orange. Blue means diapause, orange means escape, okay? So what we did is we took embryos and we exposed them to temperatures that should induce diapause, right? 20 and 25 degrees. We should get at least 80% of the embryos should go into diapause at 25 and 100% of them at 20, as you see up here. And we expose them to the various different analogs, analytes of vitamin D down the synthetic pathway. And you can see that when you get down here to the 25 hydroxy form and the 1 alpha hydroxy form, it's able to easily push the embryos into escape trajectory, even though they're in environmental conditions that should induce diapause. And I also want to draw your attention to the amount that it takes. 1 picomolar, and we didn't even try less than that. But if you look at the levels of hormones in our blood or whatever, it's usually on the nanomolar scale that people are looking at. But this is picomolar amounts. I mean, it's the tiniest amount, so they're so sensitive to it. And sure enough, if you look at those enzymes that are important for producing vitamin D, they are differentially expressed in the 30-degree fish, and you get more vitamin D in the embryos. So I think we're pretty convinced that it's vitamin D synthesis that drives them into active development. But we were kind of curious. We're like, this is such a weird thing to have DAF12 and daphrochronic acids be homologous to this vitamin D receptor. So we actually tried. We were able to find some delta-4 daphrochronic acid online, and we can actually cause the same effect with daphrochronic acid. It takes a lot more, 75 micromolar. So we have to dose the heck out of it. But it looks like the similarity between DAF12 and the vitamin D receptor is conserved enough that they can even share ligands, which is pretty cool. Okay. So our new model now for controlling diapause is that if you're under conditions where you can synthesize vitamin D, then that vitamin D will bind to the vitamin D receptor and you'll actively develop, or I would say maybe normally develop like any other vertebrate. In the absence of vitamin D, then there's no ligand to bind and it activates a completely different program that initiates diapause. And I just want to add a wrinkle to this. I don't have enough time to talk about it in detail yet, but we actually think this could be an environmental pathway too, right? Because vitamin D is generated by fungus and by plants and a lot of other things. And just think about what it would be like if you were wet mud in the tropical sun. There's a lot of fungus and other things that go in that. And then you get baked in the tropical sun, dry. You're going to get a lot of 7-dehydrocholesterol or ergosterol being converted to vitamin D2 or D3, either one will work. And then when the rains come back again, all of that basically photo damage products from the environment above is going to soak down into the soil. And so we actually think now that this might be a way to tell the embryos, hey, the pond up above you has been dry for long enough that everything's toasted. And so it's been long enough that it will reactivate you so they don't get activated by a false rain or something like that. So we think there might be both endogenous ways to make this work, but also environmental cues that can be integrated in. And I'm actually more excited about the environmental cues for some reasons. Okay, so that's that part of the story. So let's talk about another. So that is an example, I think, of epigenetic regulation being hormonally, like activating a hormonal pathway and having a different compound around, and then adding to that the potential for environmental vitamin D to activate the embryo. Another one of those pathways for epigenetic regulation is these small non-coding RNAs. So at the same time that we did that other experiment, we took those same samples, we split them in two, and we did RNA-seq on the small RNAs and the mRNA. So now let's look at the small RNA samples. So this just basically shows you that the libraries were decent. But the most important panel is down here. So here's our different developmental stages that we looked at. And probably the most important thing is just a lot of interesting information here. But what was curious to us is this MIR430. So that's this kind of blue color right here. And you can see that early in this process, there is a ton of it. It makes up almost 80% of the microRNA or small non-coding RNA transcriptome is this MIR430. And then through these developmental stages, you see that that percentage goes down and down and down. It's actually then replaced by MIR10, which is super cool too, because it helps regulate COX genes. So it makes complete sense that MIR10 would be upregulated there. But we were interested in this MIR430 because it's important in regulating early development in a super interesting way. So here's what the expression patterns look like for that. So escape embryos on this site now, remember yellow is highly expressed, blue is lowly expressed. diapause embryos here, and then this just shows where the statistical differences are between these things. But what you can see is that the escape embryos, so the embryos at 30 degrees, have a sustained and very high expression of these near 430s. And you can see that over here in line graphs if you'd rather look at line graphs. Compared to the embryos that are going to go into diapause at 20 degrees. Okay. And if you're interested in near 430 or these sorts of things, we can talk more. They've got a really cool organization in the genome, and there's all these different variants that are super interesting. But the main take-home point is that near 430 is highly expressed at 30 degrees. So we asked, what is this near 430 doing? Well, near 430 has been shown to mitigate the maternal to zygotic transition during development. So all of your early developments, for humans, it's only like till the 4 or 8 cell stage. The zygotic genome is silent. But for fish like this, and this is basically from a zebrafish study, somewhere between the 512 and 1,000 cell stage, up until that point, all of the developmental events are controlled by the maternal genome. The maternal genome is packaged mRNAs and proteins and small RNAs and hormones and everything that's needed to get you to that point. And you don't want the zygotic genome active during that time because it's going to fight potentially with the maternal genome, right? So the zygotic genome is silent. At the maternal to zygotic transition, those maternally packaged mRNAs and other things are degraded, and the zygotic genome is activated and transcription starts, okay? And MIR430 is expressed, the MIR430 expression is right here. It's expressed during that transition, and it turns out that many of the maternally packaged transcripts have MIR430 seed sites in their 3' UTRs. So they're targets for MIR430, and MIR430 targets them for degradation. And so it's a way to basically make sure that this transition is sharp and you don't get confusing information from a newly transcribed zygotic gene versus the maternal gene that is packaged. So we came up with the idea, and we haven't actually been able to test this yet, but this is our hypothesis at this point, is that there must be some maternal mRNAs that are packaged that help to induce this diapause phenotype, and that under certain environmental conditions like high light or 30 degrees Celsius, these zygotic microRNAs are upregulated and actually go and target those maternally packaged mRNAs and block them so that you can have active development. And if you look at, so we've also done some transcriptomic analysis of newly fertilized embryos. So we just let the fish spawn, we take the embryos immediately out and isolate their mRNA. And these are just the top, I think, 20 genes that are packaged into the oocytes by the mom. So these are maternally packaged transcripts. And all the ones in pink have MIR430 sites on them, okay? All of these actually have MIR430 sites on them. The ones in pink are all involved in some way, shape, or form in dormancy regulation in ways that we could clearly identify. So anyway, especially like this forkhead box, this FOXO3, which is important in almost every dormant organism that's been involved. So it looks like we have all of the parts and pieces there, but we just haven't had the time to get the experiments done to really test knocking out some of these genes or blocking them. And we're in the middle of doing that work right now. Okay, so another thing about, so I'm going to switch gears a little bit now. So now you've got a hormone story, small RNA story. Now I'm going to go back to vitamin D and talk about actually exit from diapause. So we know that we can keep embryos from going into diapause by giving them vitamin D, but we didn't know, and it isn't necessarily straightforward, that vitamin D would actually control getting them out of diapause, right? There could be a completely different mechanism. But it turns out that if we give them vitamin D when they're in diapause, that's this orange line right here, that it causes them to break diapause. So the control embryos are down here. You can't even see them because none of them break diapause. You give them, I think this was 10 nanomolar vitamin D, and they just pop right out of diapause. So we know now that we can force them out of diapause. And if you're a heat shock protein person out there, this is definitely modulated by heat shock proteins. So this blue line is if we inhibit if we give them vitamin D and inhibit HSP70, so this is an HSP70 inhibitor, it even happens faster. And then the embryos completely crap out, like they're so happy by that time, like development's completely abnormal. So we know that HSP70 works in that way, but also HSP90 inhibitor is this orange or these purple things up here. If we inhibit HSP90, we can actually delay or even prevent vitamin D from doing its job. So we know that there's interaction between the heat shock proteins and the vitamin D receptor in some way, shape, or form in the system, which is another really cool way to integrate temperature cues or other forms of stress into regulating this vitamin D specific response. Okay, so we got sort of excited about this, and we decided to do an experiment. So we're going to switch now and talk about bisulfite sequencing and look at DNA methylation. So we took embryos that were in Diapause 2. We gave them 10 nanomolar vitamin D, and it takes about 2.5 days at 25 degrees for them to wake up from this signal. And then we extracted their DNA, and we did reduced representation bisulfite sequencing, which is basically a way to look for CpG-rich portions of the genome and look for their methylation status. Okay, and bisulfite sequencing is super cool. You basically take your DNA and you sequence one group of DNA that hasn't been treated with bisulfite, and then the other part of the DNA you convert with bisulfite. Every place there's a C, it converts it to a U, and then when you amplify that for sequencing, the U becomes a T. So only the C's that have been methylated are the ones that are going to be protected and stay as C's. So you can tell the difference between a methylated and an unmethylated cytosine in this case. Okay. All right. So a couple of interesting things, and this is why you should definitely not read, if you don't study mammals, you shouldn't read a review paper on DNA methylation in mammals and think that it applies to anything else. If you look at CpG islands in mammals, they're almost always very lowly methylated and very specifically methylated. And usually that shuts gene transcription off. Well, our CpG islands are almost all methylated, like 80 to 90% of them. In fact, when we got the data back, the people that I work on lemurs and other primates, and they're like, this can't be, we must have messed something up. So we did it again, and we got the same results. So we're pretty sure that in these fish, for whatever reason, we don't know yet, they just have a super high methylation rate. So their genome is methylated like crazy. But they're still differentially methylated areas. So if we look at the data, so here's the number of DMRs is differentially methylated regions. So reduced representation by sulfite sequencing, you take the genome and you cut it up into 1,000 base pair tiles, basically. And then you ask, in this tile, how many of the CpGs are methylated, and does that percentage change in the two conditions, right? So we're looking at 1,000 base pair chunks. And what we saw when we treated them with vitamin D is that a bunch of them, 227 of the differentially methylated ones that we found, were hypermethylated, so they increased their methylation in response to vitamin D. And 54 were hypomethylated, or methylation went down in response to vitamin D. And then further, if you look at where in the gene they were methylated, there's actually a lot more gene body regulation or methylation than there is promoter methylation. And this makes the data set really difficult to interpret because we now have to go in and test pretty much all of these, because if it's in the gene, in an intron or exon, you don't know exactly why that methylation might be happening. And then there's a bunch of intergenic regions as well, which these for us, these are probably microRNA genes, but we don't have them annotated in our genome. Okay, so just a quick look at some of the things that are differentially methylated. So the hypomethylated ones, so here's just methylation difference. So 0 would be the same, minus 100% would be completely methylated or demethylated in this case. And some of the things that were differentially methylated are super interesting, and we don't know what it means yet. The most differentially methylated gene in the data set is this SLC39A1, which is a transporter that transports a plasma membrane zinc transporter. Okay. And it's a zinc importer, actually. So we don't know if the embryos are starved for zinc and they need zinc. There's a lot of transcription factors that require zinc. But we know a lot about similar ions like calcium and what their role is, but there's a lot less known about zinc and zinc metabolism and zinc signaling. So I'm super interested in exploring this. And also this CPEB3, which is a protein that is known, for instance, CPEB1 is the protein that binds to maternally packaged mRNAs and keeps them silent. CPEB3 is very similar, and it's known to bind to mRNAs and regulate translation. So those two things were highly methylated during diapause and demethylated when the embryos were activated. We're right now following up to look and see if we actually see a change in gene expression, or more importantly, in gene activity in those. Okay. So there's just a bunch of other really cool things. Oh, the other cool thing about this is the gene has a ribozyme in it, the hammerhead ribozyme, which is super cool, but I have no idea what it's doing yet. Okay, and if we look at the hypermethylated genes, a whole bunch of different ones, we oftentimes see things like endosome cycling is super important in the transition from active to dormant. And there's this laminin B1 nuclear envelope gene, which is super important in regulating the structure of the genome, which is another probably epigenetic issue. And then the one that I like the most is this ID2, which is a zinc finger its whole job is to go and find other zinc finger transcription factors that stick into their zinc fingers so they can't bind DNA. So it's a way to like globally shut off zinc finger proteins. And the genome of this fish has like 800 zinc finger protein transcription factors. It's pretty crazy. So I'm pretty sure we're going to find some interesting stuff about this, but we haven't really delved into it yet. Okay, so there's a story about DNA methylation. And I just want to give you, so that one was in response to vitamin D. I want to give you a quick look at what happens in terms of embryos exposed to stress and DNA methylation. So, of course, the most important things for these fish to survive the dry season is they couldn't survive being dry, right? You got to survive without water. And they do this, it's super interesting. So here's a Diapause 2 embryo, there's the embryo. There's this perivittling space between the embryo and the edge of the chorion. So fish eggshells are made of protein. In this case, in annual killifish, the protein coat is like 25 micrometers thick, super thick. For something like a zebrafish, it's like 1 micrometer thick. So they've got this super tough protein coat. When you dehydrate them, and we just set them on super easy, these are the best experiments to ever do. It's really easy to control relative humidity. You can take a saturated salt solution and put it into a desiccator like this, and you just put the embryos on filter paper in the air phase over that, and the saturated salt solution will control the relative humidity in that confined space. So we can do 75 or 85% relative humidity. These studies were done at 85% relative humidity that I'm going to show you. But here's a dry embryo. So this embryo has been sitting on a filter paper in the lab for, I think this one was for a year. And it sits just like that. This is actually on a glove. That's what the black background is. So they're amazing because they can just sit there, their heart beats slowly, and they just kind of slowly develop while they're dry. So this graduate student of mine, Daniel Zajic, did this work a couple of years ago. And just to show you their ability to survive, oh, whoops, I didn't update this slide. Sorry about that. This slide should have been out to 600 days because he actually did finish this and get his PhD. So the LT50 for drying out at 85% relative humidity for a Diapause 2 embryo is like 600 some odd days, whatever. So anyway, way long enough that they can survive from one season to another, right? All right, so we did a metabolomics analysis of this, and we used metabolome to do it, which was both good and bad. I don't know if anybody else in here has worked with metabolome, but you get a lot of data, and some of it's hard to interpret. But just to give you an idea, here's the graphical representation of the data that we got for an embryo that was 4 days post-diapause. So it's developing again, it's not in diapause. And they can survive for probably 200 or 300 days like this. And we just took them at time 0, so they were wet, and extracted their metabolites. And then after 18 days of being dried out, and extracted their metabolites and compared the two. The red circles are things that are upregulated, the blue circles are things that are downregulated, and the size of the circle tells you not the amount but the significance of that differential regulation. So you can see there's a lot going on, right? There's a metabolic map, and there's a lot of different things going on. What I want to draw your attention to today, just real briefly, is this TCA cycle, which is in the middle of this right here. And one of the metabolites that seems to accumulate in huge amounts during dehydration is 2-hydroxyglutarate, which is not a normal byproduct or not a normal intermediate of the TCA cycle, right? And it's generally generated by either mutant isocitrate dehydrogenases, or in some cases, LDH will actually produce it in certain situations. But it's very similar to alpha-ketoglutarate, and that's why it shows up on the metabolome here. And so we see this huge increase in 2-hydroxyglutarate, not something that we expected to see. So we looked into it, and we got super excited when we looked at this, because it turns out that 2-hydroxyglutarate is considered an oncometabolite. And in a number of different cancers, they find it is highly upregulated, and it screws up epigenetic regulation because it is a competitive inhibitor for alpha-ketoglutarate dependent dioxygenases. And these alpha-ketoglutarate dependent dioxygenases are things like Jumanji and other things that demethylate either histones or the genome. So this is a way to directly link the metabolome to the epigenome, which I think is super fascinating. And so by environmentalists stressing them, somehow they're producing this 2-hydroxyglutarate. We're not sure how they're producing it yet, but we're working on that right now. We basically have to use NMR to look at an enantiomer that is produced, because different enzymes make different enantiomers. And once we figure that out, then we'll know. So, so we're looking into this. So we did a quick experiment with this, where we took Diapause 2 embryos, we broke them at 4 days post-Diapause. We treated them either under aqueous conditions or 7 days of being dehydrated, and then did bisulfite sequencing just to look and see, does anything change? And sure enough, we see huge changes in the metabolome, or sorry, in the epigenome. So there are, again, in this case, it's just the opposite of what we saw with vitamin D. There's a lot of hypomethylated genes in response to dehydration, and then a few hypermethylated ones. And this work, we haven't really gotten much farther than this yet, because we're still right now in the process of trying to figure out what it means to, if you're methylated in the gene or in the promoter and all those other places. And I don't want to really publish it until we kind of have a better idea for what the spot of that methylation means. But lots of really interesting genes are differentially regulated in this case. Okay, so that's it. I want to thank my collaborators at OHSU's Lucia Carbon's super awesome epigenetics person that helped with all the methylation analysis. I've had funding from a number of different sources. And of course, I'm not in the lab much anymore. So these are the graduate students, mostly Amy Romney and Daniel Sajik, that did the work that you saw today. And there's also usually an army of undergraduates in the lab helping take care of the fish and do experiments and stuff. So I'm gonna, all of these folks participate in these studies, and I'm happy to take questions. Yeah.
That was extremely interesting. One of the findings that was really interesting was the conservation in entry to diapause in killifish and entry to jowler and C. elegans. And you had a number of findings later on about diapause, such as how exit from diapause happens. I was wondering if you looked at any of those, such as how exit from jowler happens, C. elegans, if there's any conservation there as well.
Yeah. So up until recently, there hadn't been a lot of information about exit from jowler and C. elegans, but there was just a paper this year, and I was mad that I was sitting on this data, because they came out with the same thing, is that difroconic acids will boost them basically out of jowler. So it looks like it's the same hormonal pathway going in and coming out, and it's the same for us. But well, I mean, I shouldn't feel too bad about being beat by C. elegans, right? Yeah, more.
Thanks, Jason. Your MER430 data and its ability to silence the maternal genomic effects, why doesn't that also silence the transcriptome of the
Because the embryo, so the embryonic, so the way the microRNAs work, right? They've got a seed sequence and a 3' UTR typically, although they could be in other places. The embryonic transcripts that get expressed don't have that seed sequence, so they're not targeted by the MER430. It's really the ones that are packaged by the mom. And this is why, I don't know if you've heard of the concept of epigenetic programming, right? So, or imprinting is sometimes called. So certain genes have to be expressed from the maternal genome or the paternal genome during development. And it may be that the reason why is because the maternal genome expresses a variant that has a UNLV 430 response element in it, or sequence in it, and the paternal one doesn't, or something, right? So it could be something like that. But yeah, if you look at the zygotically transcribed genes, they don't have your 430 sequences by and large anyway.
Yeah, these fish, they resist a lot of environmental stresses, right? Like the network environment. Does the escape have any relationship with resilience or resistance for these environmental stresses later in life?
Yeah, so it's interesting. We're doing a study right now to look at what the differences are between embryos that go into diapause and come out versus the ones that escape diapause, to see if there's, we haven't tried stress resistance. We're looking for like lifetime effects, like do they age differently, do they have different fecundity, things like that. But stress tolerance is really high in all of these embryos. So for instance, the reason that we did that one study with the 4-day post-diapause embryos is that despite the fact that they're actively developing, they have the same anoxia tolerance as a diapause embryo. So in fact, they're actually slightly better at anoxia tolerance than a diapause dormant embryo. So there's a window after the embryos break diapause of about 4 to 6 days where they retain that ability to have high stress resistance. And then it starts to fade off. By the time that they finish development, they're sort of like a typical fish. They're not that tough anymore, although fish are pretty tough in general. Yeah.
So it was interesting to me, you were showing the amount of difference in methylation of different genes, and I think it was the SLC39A or something like that that showed the greatest difference. Do you ever see clustering, because of the genome duplication events in fish? Do you ever see clustering of like, it was the only SLC that I saw.
Yeah.
For, okay, where's the SLC 585? It's
Yeah.
not in that list. You'd think that they would be methylated at the same rate within these orthological parallels.
Yeah, we haven't done, we haven't done a detailed analysis of the structural relationship of these differential methylations yet, because our genome, so we have a draft genome sequence, but it's still got, I don't know, 100,000 contigs or something. So the spatial organization of the genes are not clear to us yet. I mean, they're probably very similar because synteny, like the organization of genes and genomes, is usually pretty conserved. But the amount of genome duplication in this species compared to, for instance, zebrafish is pretty low. So they have, the cyprinids and zebrafish, for instance, went through that extra genome duplication. It's still pretty new. Their genome duplication was a long time ago. So for some genes, they still have multiple copies. For instance, for the vitamin D receptor, they have two. They have an A and a B. And the A is the one that's most like the one that we use, and B is the one that's expressed during development and is used there. But for lots of other proteins, they've lost those other isoforms. So I think the total genome size for these guys is right around 1 gig base. And 46% of that is repetitive, which is why we don't have a better genome. So, hey.
Okay, so I think we're good. So what we're going to do now is